The broadcast is now starting. All attendees are in listen-only mode.
Good morning, everyone. Thank you for attending this GenScript sponsored webinar today entitled A Novel Endotoxin-Free Microbial Recombinant Protein Expression System, Solutions for High-Quality Proteins. My name is Dr. Shalise Ammons, and I'm a marketing specialist for GenScript USA, and I will be helping to moderate today's webinar. Just as a reminder, if you could please mute all your microphones during the presentation. If you have a problem or if there's a question, please place it in the chat box or the question box, and one of the moderators will see and address your question. At the end of the webinar, we will be taking additional questions. However, since time may be limited, we may not get to your question today. Therefore, we will record all the questions, and we will then email a response to you individually. So without further ado, I will go ahead and introduce today's speaker, Dr. Brady Wu. Dr. Brady Wu graduated from Peking University with a Bachelor of Science degree with an honor in Biological Sciences in 2007. He attended Ohio State University and received his PhD in chemistry in 2013. In the same year, Dr. Wu joined GenScript as a senior scientist in the protein department. In 2014, Dr. Wu became a group leader of the project management group and was responsible for managing project managers and senior scientists. In 2018, Dr. Wu was promoted to the director of protein department and currently manages over 100 employees. He is also the strategic head for the business development and the financial planning for the same department. So with that being said, go ahead, Dr. Wu. You can go ahead and get started.
Thank you very much for Shalise for your very nice introduction. So today's title is a novel endotoxin-free microbial recombinant protein expression system. I think it is a very good solution for high-quality proteins. So the talk will be divided into two parts. First one is introduction of GenScript and our services, especially our protein capability, and the second part will be the expression system we have developed in the last few years. So GenScript is a global bio CRO. It was founded in Piscataway, New Jersey, USA in 2002. In the end of 2016, we went public in the Hong Kong Stock Exchange market. And right now we have well over 2,000 employees. We are a top company globally for gene synthesis and a top bio CRO in China. So we have our production site in US, Nanjing, and Zhenjiang, Jiangsu province in China, and we have local sites in Leiden, Europe, Netherlands, and Japan. So we are a truly global company. We are offering one-stop service. We are specialized in biologics discovery and development, along with high-quality reagent service. We have local technical support, and we have a 24-hour customer service. So whenever you want to talk to us, we are available for you. We have fast global logistics. We can ship all over the world, from US to UK to Europe, even Africa and South America. And we have a competitive pricing, and we are very stringent on the intellectual property protection for our customers. So for track record of GenScript protein, we have been in the protein business since 2004. The department has a very comprehensive portfolio for producing proteins, and you can see in the figure on the right, we have been producing more than 10,000 of high-quality proteins with the success rate more than 95%. We delivered more than 95% of the proteins requested by our customers, and we have done varieties of proteins, including transmembrane protein, antibodies, nuclear hormone receptors, growth factors, and cytokines. Right now, we are carrying 4 systems. The major systems we are using are bacteria, insect, and mammalian. We also have a small platform for yeast, but it's more like an industry-used platform. Our customers are more on the R&D purpose. So these three systems are coming from different organisms, obviously. Bacteria, we normally use E.coli, and for insects, we use Bacillus system. We use cells such as SF9 and Hi5, and we use virus to infect the cell to produce proteins. In mammalian, we use CHO cell and HEK cells. Both are suspension cells, which are very easy to scale up, and we can do very large extraction volume in all three systems. For bacteria, we have a fermenter go up to 500 liter one batch. For insect and mammalian cells, we have a well bag reactor available, and we also have a single-use bioreactor available as well. So obviously, these three systems will have different purpose in terms of R&D uses. For example, for bacteria, we mainly use that for antigen, especially for simple detection purpose, such as Western blotting. But on the other hand, it's a very good system for intracellular target proteins, especially for structural analysis. For example, if you want to do a transcription factor, probably bacteria will be the first choice. We can also produce a lot of true enzymes. I can show you some figures later, such as polymerase, Cas9 series proteins for gene editing. And overall, it's a very cost-effective system. It has fast turnaround time, but sometimes you got low protein solubility and you have in vitro folding issue, and you need to do refolding. And sometimes you have to fight with the high endogenous endotoxin level from E. coli, which could be harmful for your assay. The next one is Bacillus insect system. It's especially good for producing kinase proteins and complex. And I can show you some of the examples later. We have done a lot of high quality proteins for structural analysis and also functional analysis. For mammalian cells, you might be familiar with stable cell line and transient, who are especially good at transient expression. We can produce a lot of recombinant antibodies in high throughput fashion, and also some extracellular target proteins in human or animal, from human or animals. So each system has its pros and cons, and so I'll show some of the examples we have. This is from E. coli bacterial system. We have done a lot of gene editing related enzymes, and also some enzymes used in molecular biology process, such as T4 ligase and polymerase. Overall, we can get very high quality proteins. For this one, you can see it's a very large one, but we were able to obtain tag-free protein, relatively low yield, but very high quality. And it's also with low endotoxin level, so it's good for cell-based assay if you want to do gene editing in cells directly. And the next case is a kinase in insect. We have done significant work of kinase, especially for structural studies. So from 10 liter of F9 cell, we can generate about 100 milligram in total. That will be good for several batches of structural analysis already. And this protein was labeled and is easy degradate, but with multiple purification schemes we mastered, we were able to get high purity protein. And the insect system is also good for multi-subunit complex. We have done 5 subunit complex 4 subunit complex and also 3 subunit complex, because you can use different virus ratio to infect the cells. So that way you can optimize the expression. Sometimes you have high expressors for one subunit and low expressor for another subunit. But for inside cells, you were able to tune this process and get the optimized ratio and optimize the assembly of your target complex. And we also have done several very big molecules in mammalian. So IgM and IgA molecules, antibody molecules, which can be used in diagnostic assay. Sometimes our customers ask us to produce this kind of difficult molecules. For IgM, you can see from reducing the reducing assays page, you can see about 4 bands, 3 bands. The light chain, heavy chain, somewhere there is a J chain. And the non-reducing, you have a very big molecule which doesn't migrate well on the non-reducing SDS page, but it's functional, and we have a lot of expertise in expression and purification of this kind of big molecule. We have to tune on the plasmid ratio, and we have to choose the best cell line and the best purification scheme to get high purity IgM protein. The red one is IgA protein, and you have actually 4 chains: light chain, heavy chain, IC chain, and also J chain. One more chain than the IgM molecule is a dimer. IgM is a pentamer, and we were able to get functional IgA protein. And of course, we have done a lot of other molecules such as recombinant antibodies, and that is becoming a kind of routine work for us. But we also have a lot of expertise dealing with protein complex in mammalian cells. Okay, right now I'm switching to the main topic of today. That's the introduction of our new system we developed this year. It's Bacillus subtilis. Unlike E. coli, it's a gram-positive bacteria, and I'll show you what will be the difference. It also have a rapid growth about as fast as E. coli's platform. Unlike E. coli, it has very powerful secretion capacity, and also it's a food-grade microorganism. It's considered as grass, which means generally recognized as safe. So we think it's another excellent bacterial expression host. So compare these two bacteria. For the gram-negative bacteria such as E. coli, you have a cell membrane, or we call inner membrane. We also have outer membrane, and it also comes with LPS, which is endotoxin, which is harmful for our body, harmful for the cell's growth, and it's not good for the cell-based assay. It's not good for animal work. But for the gram-positive bacteria, you only have one cell membrane. You don't have LPS. That means if you have some protein flow to the out of the cell membrane, it will be secreted into media. But in the case of E. coli, the protein will be secreted to the periplasm first. So it's a very good secretion system compared to E. coli, and it doesn't have any endotoxin. So what about the production workflow? It's quite similar. We have the cell stock, and we make committed cell, and we use transformation, put DNA and the committed cell together, and we do clone selection on the plate. So we have all of the single clones, and we do shake flask culturing. So it's quite simple. It's just the same as E. coli process. So we show some bottom-up approach when we're building the system. So first, we want to see whether we can build up an intracellular expression system. I didn't mention earlier, but Bacillus is a very extensive toolkit. We have a lot of different options. At least in this case, we tried the origin of E. coli and origin Bacillus in the same vector, so it can be replicated in E. coli and Bacillus. And we also try the promoter, 3 different kind of promoter. One is controlled by lactose operon. The other is controlled by cellulose operon, and the first one, the last one is controlled by nothing. It's a constitutive vector. So also we choose different copy number in Bacillus. One is low copy number, the other is mid copy number, and the third one is high copy number. So again, it's already a lot of selections, but we just want to see whether we can get a system going. So we choose a favorite target, it's GFP, and so we can see on the SDS page all 3 intracellular systems showed a good expression of GFP, and the high copy number system showed a very high level expression, shown here. So that one is working. So next step, we want to produce extracellular expression system. As I said earlier, the Bacillus system is a very good system for secretion expression. So we put some signal peptide after the promoter. One is amylose E, and the other is BPR. The third one is NPRE. So we produced another other 3 vectors for extracellular expression, and we choose a targeted protein PGL for expression. Unfortunately, the high expression vector, high copy number vector with the signal peptide didn't work out. But for the low copy number secretion system and middle copy number secretion system, we have the protein expressed in the media. So it's a model system, but it's quite neat because we can directly purify the protein from the media. Unlike E. coli, you have to use some lysis method specifically to get the protein out of periplasm. So it's a very big benefit for us. So we want to see whether this system can work on some real targets, not just more protein. So we try the purification of GFP because we say, okay, this system is good, and we want to see whether we can get the benefit of endotoxin. So we tried a normal process without any endotoxin control on our instrument, our solution, and we tried a more stringent condition with good control and also a step of endotoxin removal. But the endotoxin level is already lower than 10 EU per milligram for the case of GFP without any endotoxin removal operation. So we think it's a very good system because for this system, we can get low endotoxin protein from a bacterial. So that's a very big benefit. And say, okay, how about we can try some others? So we try some VHH, which is a single domain antibody, and of course with his tag, it's kind of antibody fragments, and we say, okay, whether we can have this kind of antibody fragments segregated into media, and whether we can get them with low endotoxin, and it was very successful. All of the VHH have a relatively reasonable yield from 5.5 to 7, 3.8 to 7, and all of them have very low endotoxin level. It's lower than 10 EU per mg, and sometimes it's already a level good for animal model study. Again, it's without any endotoxin removal. Okay, we'll say, okay, we can do model protein, we can do which, how about some cytokines, because cytokine can be produced in bacteria. A lot of vendors are selling cytokine from bacteria. But for cell-based assay, we'll see whether we can control that into a very low endotoxin level. So we do a comparison study in E. coli and Bacillus. It's L2 for this protein, E. coli and Bacillus have this about the same yield, 2 and 2.5. But Bacillus again showed very low endotoxin level without any endotoxin control. For the E. coli, if you want to reach endotoxin level to about 25 EU per milligram, you have to do extensive endotoxin control. So, and again, it's a very big benefit for this system. Okay, so I will say we have that kind of benefit, and if we want to do some other proteins, what Bacillus can do for the proteins E. coli cannot do. So we tried two very difficult proteins. So the left two lines are STS-page, the right two lines are Western blotting, and fortunately, both proteins showed better expression in Bacillus system. So that actually will give us a new solution besides the benefits. If we have some difficult proteins, probably Bacillus will be a different option if the E. coli didn't work out. So we did a very simple comparison between Bacillus and other expression system. First one is gram-positive, and we can do intracellular and extracellular expression. So it's quite a diverse platform already, and it goes with the same short period production and low cost compared to E. coli, but we really think it can give us better quality protein for the application, and it is not very difficult to work with Bacillus system compared to insect and mammalian. So we really think it's a very good supplement for our arsenal, and we hope in the future we can give our class more benefits when using this system. So to summarize this short talk, we have developed a Bacillus subtilis expression platform for producing recombinant proteins. So we have three intracellular expression vectors with low, middle, and high expression levels. And we also have three extracellular expression vectors. And again, if low endotoxin is necessary, Bacillus could be a good option. Actually, this is a second webinar talk I did this year, and the first one is also on our R&D efforts. So with our scientific expertise, I really believe you can trust us to provide your research materials faster and with the highest quality. And in the future, we will continue to improve the capability and to offer you best reagents for life science research and also drug discovery. And with your help, we'll make research easy. And that will be the end of this short talk, and I will hand back to Charlize.
Hey, Bo, thank you very much for your talk. It was a very good presentation. So again, everyone, my name is Dr. Charlize Ammons, and I am the marketing specialist for GenScript USA. So I will just be giving a brief introduction on some of the protein services that we went over today, and also mention some of our key service features. So while I'm doing that, please go ahead and type your questions in the question box now. So as Bill mentioned, protein services, we can, with our technical expertise, we're able to really provide our customers with the best service. for a very cost-effective option. So just to begin, I would like to talk about first our workflow. So for all our protein services, we do start with a customer-provided sequence, or we can start with the customer-provided DNA. Then the sequence goes through our own codon optimization, which is a proprietary codon optimization service where we will do gene synthesis included in your protein expression. Then we will clone your sequences into a construct that is, again, one of our proprietary constructs. And then we will do cell culture and transfection. We will do protein or antibody purification, and we will also provide QC analysis. And then we will deliver to you purified recombinant proteins or antibodies. So just highlighting again our key features. Some of our services are guaranteed, and that guaranteed means that we will guarantee you an expression, protein expression or antibody expression in yield, purity, and endotoxin level. Again, we use our proprietary codon optimization and expression vectors to provide you with the best gene synthesis and expression, and it's included at no additional cost. We can also guarantee the delivery. So for some protein projects, we'll be able to deliver within 4 weeks. It just depends on the service that you're ordered. And then again, some projects have a no charge if the project is not successful, which will be outlined in the proposal. Next slide, please. Thank you. So just a highlight of some of our key services. One is our back power, which is our guaranteed expression service in the E.coli system that Bill mentioned earlier. Our manpower, which is guaranteed expression in our mammalian system for protein and antibody expression. And last but not least is our man pilot. It's a new service where we can guarantee antibody expression in a mammalian system. This service can also be ordered as a pilot expression for different scale-up projects down the road. And if you have any questions, please feel free to contact us at protein@GenScript.com or the phone number that's listed at the back. So thank you everyone very much, and I will go ahead and begin with questions. Okay. So Dr. Wu, our first question is, what is the major difference between E. coli and Bacillus, and what are the advantages of the Bacillus system?
So E. coli, in the history, E. coli is a mostly used system. But on the other hand, Bacillus was not widely used in the R&D purpose. It was more used in the industry purpose because it was considered as a grass organism. But I think for the research purpose, the Bacillus still have an advantage of different options of expression selection. For one strain of Bacillus, you have more than 100 different signal peptides and different promoters and plasmids you can use. And also, in nature, it can produce high-quality proteins in terms of low endotoxin. So I think that will be a very big advantage of the Bacillus system.
Okay, thank you very much, Dr. Wu. Our next question is, what could be the future development of the Bacillus system?
For now, we just got started, so we really want our customers to let us try the difficult projects or the projects we think are suitable for the Bacillus platform. In the future, we will see what will be the best option for each category of the protein which can be done in Bacillus, for example, for enzymes or for antibody fragments. We'll optimize the system application oriented, and for now it's really a bottom-up approach, and in the future we want to know more what system can do. I think that's very important. Sorry.
Thank you very much. Our last question today is there a specific product category you are looking for by utilizing the Bacillus system?
The first one will be antibody fragments. As you can see, we can get low endotoxin antibody fragments very easily, and that can be good for cell-based assay. And it comes with a very big advantage. And we are looking for the enzyme production as well, because E. coli is good for enzyme production. I will say Bacillus will have the same kind of characteristics compared to E. coli. And also, Bacillus is a very good display system, actually. And so in the future, if we want to do some kind of protein engineering project, probably we can develop some display system of Bacillus.
Thank you very much, Dr. Wu. As I mentioned, our time is limited today, so any additional questions will be recorded by the moderators and will be answered through a personal email. I would like to thank everyone in the audience for your attention and participation in today's webinar, and I would like to give a special thank you to Dr. Wu for coming on and speaking with us today about the Bacillus system. Thank you, Dr. Wu. Okay. And thank you everyone for your participation today.